SMA stands for spinal muscular atrophy. It used to be the leading genetic cause of infant mortality. It's a genetic disease that affects approximately 1 in 10,000 newborns, and it results in the progressive loss of nerve cells and motor neurons that allow muscles to contract. At birth, the patients may look normal, but within some period of time, they don't achieve the developmental landmarks, like, for instance, the ability to sit that a child gets, some months after birth. Or in some cases they can do this, but they cannot stand, or if they're able to stand or walk, then over time they lose the ability to do that. So there is a progressive character of the disease. And so there's progressive muscle weakness, there's respiratory symptoms because of weakness of the muscles that are necessary for breathing. And so patients are more susceptible to respiratory infections, and so there's a collection of complicated symptoms. Some are motor problems and some are respiratory problems. With the therapy for SMA, Nusinersen or Spinraza, the effects can be very dramatic. This is considered a disease-modifying therapy, and the magnitude of the effects depend on when the treatment is in relation to when the disease starts. So if in the best case scenario, the treatment can start very shortly after birth following newborn screening, and in that case, the treatment can prevent the onset of the disease. So even the most severely affected patients in terms of the genetics will now not exhibit the symptoms, and they can develop motor functions normally, so far for up to 5 years and hopefully much longer. If treatment is done later, after the onset of symptoms or after the disease is more advanced, it can still slow down the course of the disease, and it can allow for not just stabilization, but improvement of motor functions, even for patients who are already severely affected. Our work on the antisense oligonucleotide for SMA was done in collaboration with Ionis Pharmaceuticals. So it brought together an academic lab and a biotechnology industry. And the initial observations that were made in my lab began to give us a hint that it is possible to augment exon inclusion with an antisense oligonucleotide. I think conceptually it wasn't obvious for us, but we made an observation as part of more complicated experiments, and then we realized, A, that it's possible, and B, we could probably optimize and get more potent molecules that do this. And so then in order to do that, we began to collaborate with Ionis Pharmaceuticals in 2004 to systematically screen and find antisense oligonucleotides like that. This ASO for SMA, Nusinersen or Spinraza, broke new ground in several respects. I mean, for one thing, it was a first therapy for a neurodegenerative disorder. It's what's called a disease-modifying therapy, because it really is changing the course of the disease in a favorable way. And it was the first time that an ASO was used to fix a splicing defect that led to a drug. Also another sort of milestone in this process was showing for the first time that if you treat a neurodegenerative disease patient pre-symptomatically, in this case for SMA, you can actually prevent the onset of the disease, and that was not shown before. So the splice-correcting ASO was developed on the basis of fundamental knowledge about RNA splicing mechanisms. So before we could even think about how to develop an ASO for this disease, we had to understand what is wrong with the expression of the SMN2 gene compared to the SMN1 gene, what happens to the RNA, So these are all basic observations. And then even though it seemed clear that the SMN2 gene would be a logical therapeutic target to increase SMN protein levels, it wasn't obvious how to achieve that. So we had to study the splicing of exon 7 of SMN2, understand why it's inefficiently recognized, what are the reasons for it. And only then we could kind of conceptualize an antisense oligonucleotide, and then we could try to design and screen and optimize for such a molecule. As a basic scientist, we get, of course, a lot of pleasure from seeing research becoming successful, but the fact that in this case it led to a drug that can help thousands of patients gives it a completely different dimension. And I've had the pleasure to meet and become friends with a number of SMA patients and their families. And so it gives me a very warm feeling, and I think the same is true for my collaborators, my colleagues in this project, that the project went all the way from the bench into a disease-modifying therapy that helps people.